So I think we are now ready to start. So I will start by saying good afternoon and a great welcome to everyone to this kickoff webinar. And it's with great pleasure that we enter the sixth edition of Food for Sustainability Agriculture's series. And as you see in the screen, our first track is Soil Microbiome, the Farmers Productivity Foundation. Throughout January, we'll have this topic, and we will bring to you different perspectives on how soil microbes are indeed a foundation. And today we want to move a little bit further and actually think that microbes were also the foundation of life on Earth. They were among the first forms of life, and they still act as the planet's primary engineers. They transform minerals, they cycle carbon and nutrients, they build soil structure, and they, of course, create the conditions that allow the plants, the animals, and ultimately our farming systems to exist and to remain productive. And this is precisely the topic that we bring to you today, microbes as the soil engineers of fertility. For those that don't know me, my name is Claudia Costa. I'm the director of Food for Sustainability Academy, and I'm truly delighted to guide you through this journey from January to May, every Thursday, as we explore cutting edge topics like the one that we start today, soil microbiome, but we'll also have digital tools, next generation agriculture, and EU policies for regeneration. And I will start also to give you a brief background about food for sustainability. We bridge research, education, and practice in order to accelerate the transition to more resilient and regenerative agri-food systems. And the key pillar of our activities is our soil health and function. This facility is powered by a state-of-the-art molecular biology laboratory, which was established in 2023 and is located in North Lisbon at a place called Arruda dos Vinhos here in Portugal. And this facility and its committed team, they deliver soil microbiome analysis, so the profiling of bacterial and fungal communities, and they also perform target microorganism detection and enzymatic activity analysis. And these insights, they actually give farmers actionable insights to optimize nutrient cycling and, of course, crop resilience. So if you want to know more, please do not hesitate in getting in touch. And we also have our pioneer integrate solution. This is called the Ampliaqua, which is a multi-trophic system that combines the microalgae, the fish. You cannot see the fish in the image, but you see the fish tanks, and also vegetables. And this is all supported by an on-site bioreactor that allow us to provide tailor-made microbial inoculants that actually close this nutrient loop and reduce the need for synthetic inputs. And we are also proud to present you uh, um, a pioneer project. This is the Aidanha Food Park. There is indeed a unique and novel approach here in Portugal, and it tries to recognize the strengths of rural areas by valorizing its sustainable game meat, unique autochthonous species, and the silvopasture systems. And this project designs regenerative protocols for pasture, and it delivers direct impact on meat quality. We are talking about things like the levels of omega-3, antioxidants, and of course, very important, the optimal ratios between omega-6 and omega-3. And these parks main pillars invite us to think if meat is all the same. The old saying is not the cow, but it's the how. And it includes things such as a slaughter or processing unit for traceability and premium market access, a circular agro-industrial hub that wishes to minimize waste through the production of biofertilizers and energy recovery, and also the economic revitalization of a new value changes that chains that are actually geared and designed to boost local sustainability and export potential of these regions. And these infrastructures, they indeed support the work that we do here at Food for Sustainability, which is to democratize soil microbiome knowledge, turning this knowledge into actionable insights that truly deliver value to our primary sector and allow a stronger positioning for our farmers across Europe. But just before we dive into today's topic, let me just very, very quickly introduce you to our excellent speakers. I'm really thankful for your availability and being present here. So we will start with Ieva Erdberga. She's a Latvian soil researcher and educator focusing on nutrient management and sustainable land use. This will be followed by Rocío Barros. Rocio comes from Spain, more precisely from the University of Burgos. She's a professor on microbiome solutions for soil restoration, and she will showcase some of the EU projects that she's been delivering, such as the Tribiome and the BioRem. And finally, we have the pleasure to have with us Ademir Calegari. He's a well-renowned EAPR research. Ademir has more than 40 years of experience on the ground, mainly on cover crops, soil microbes, and regenerative farming systems. Okay, it's again a true pleasure to have you here with us. If you want to know a little bit more about the background of our speakers, there's everything in our website. You can just have a look there. So just before we proceed, now I'm just going to set some house rules just to ensure that our session runs smoothly. So questions are of course very welcome, but I will ask you to place them in the chat here in Teams. I will direct your questions to the speakers at the end of each presentation. So we'll have a short clarification period after each presentation, but our goal is to leave 15 minutes at the end with the three speakers for a round of Q&A. We also have certification for those that wish to have a certificate, for those that complete a brief quiz that the team will give you at the end of the session. And in this edition, we went a little bit further and we have prizes for those that are fully committed to our webinar series. And we will, at the end of the series, make sure that you are eligible for these prizes once you have completed our brief questionnaires. One final point. Our webinars are recorded. They are available on our website. So if you are uncomfortable with a recording, please just switch your camera off, and we will, like this, we'll make sure that you not appear in the recording. Okay, I think this is about it, about but we can learn the rules as we go along. So I think it's time for a warm up. I think the team now has prepared something for us to warm up in terms of our topic. To get into and to dive into concrete examples and not just theory. So I will ask the team to share the screen. The screen is shared. Apologies. So let's jump into Slido. Okay. So you have here the QR code. You can just open Slido through the QR code. I think the team has already also placed the link in the chat. This is very easy. You know that you have a box. You just enter this code, hashtag F4S Academy. Let's dive into the questions. So what has the team prepared for us today? Let's have a look. Okay, and the first question is, what percentage of the world's species are found in the soil, according to recent studies. So we are talking about how much biodiversity, in terms of the entire biodiversity in our planet, is directly dependent in the soil. Okay, so the voting has started. Okay, we wait for your answers. Now, I'll just give you a brief explanation. So, recent studies on global soil biodiversity indicate that a large proportion of Earth's species are indeed associated with the soil habitat. So, by compiling and integrating, data among the major taxonomic groups that include things like bacteria and fungi, but also plants, invertebrates and vertebrates, research have estimated that approximately 59% of all species on Earth live in the soil. Okay, we know it's a very big percentage, but not as high as you suggested. But this means that this species spends parts of their life or their entire life cycle in the soil, or they are just dependent on it ecologically. So again, this shows the importance of the topic that we are addressing here. Also, make sure that the others around yourself know how important soil is and how much biodiversity it contains. Thank you so much. I think we can move to the next one. So, do you have an idea how fast can soil bacteria multiply under natural soil conditions? Are we talking about roughly every half an hour, every few hours to days, every year, or they never multiply in natural soil conditions? And this points us to an important issue. Some of you are already aware, but the growth dynamics in natural soil indicates that soil bacteria normally they reproduce at a slower rate than what we idealize in our laboratory cultures. So in contrast with the rapid doubling of times when we have nutrient rich culture media, so when which is normally around 20 to 30 minutes. So this is in the lab. Bacteria generation in the soils are constrained by resource limitations, temperature fluctuations, moisture variability, spatial variability within the soil matrix. Okay, so in the lab, we can have this multiplication of bacteria of around half an hour, but in average good conditions of a soil, it takes a few hours to days. Okay, so well done. They multiply fast, but it's not as fast as in the lab. But this also shows the potential for us to manage well the bacteria that happen in our soil, that are present in our soil. Thank you very much. You're doing very well. Thank you for your support also. And one final question. Are you aware what percentage of the carbon stored in soils is directly regulated by microbial activity? So we have here four options. Is it so the carbon that is stored in the soil directly regulated by microbial activity? Is it about a fifth of this carbon? Is it half of the carbon? Is it 80% of the carbon? Or is it all carbon that is stored in the soil responsible is the responsibility of microbial activity? And again, recent studies have looked carefully into this matter and show that soil microbes indicate indeed a substantial portion of global soil carbon is regulated by microbial activity. And this is measured by integrating major microbial groups such as bacteria, archaea, and fungi. And roughly, the absolute number is that microbial activity is responsible by the storage of 2,500 gigatons of carbon. And this carbon in the soil is transformed, destabilized, or is released through microbial processes. So it's a really huge amount of carbon. And in terms of percentage, this is 80% of the carbon that is stored in the soil is directly regulated by microbial activity. Again, this shows us the importance of understanding these microbes, of looking after these microbes, and ensure that we know and to respect their work. Okay, so thank you so much. I feel much warmer now. Thank you so much, guys, for these questions. So I think we are ready to start. So let's go. Yeva, hello. Um, I will
Hello?
now call Yeva to the floor. It's a great pleasure to have you here. Yeva, you can share your screen whenever you feel ready.
Yes.
Yeva is going to tell us a little bit about biostenosis. Okay, let's hear about biostenosis, Yeva. Thank you so much.
Hello. Everybody
Yes,
see me? Everybody hear me?
all good from this end.
Very nice. So hello to all. My name is Ieva, and um, I'm from Latvia, quite far from your place, and that sometimes gives me perspective to look at some things a little bit differently. So, what are we going to do today? We are going to answer on the most important question in our lives. What is the most important thing on this world? And of course, I can answer already. This is a soil. And we can further, let's say, remind us some golden rules of soil and how it happens to be. So one of the main thesis is that soil is the basis of biocenosis, and that is true. And sometimes it somehow is misunderstood. We as a scientists sometimes see on things very narrowly, like guy who looks on the birds, he thinks only of birds. Somebody let's say works with plants and it sees only plants, and then there are people who works only with microorganisms and they see only microorganisms, and they think that everybody thinks like they, and in these pictures they are sometimes misled about whole picture. So the often farmers think that if they get very good soil, which is rich in nutrients and then in organic matter, that is enough for good farm to be in this place for 100 and maybe for millennia. But that is misunderstanding. Of course, biocenosis is the reason why there is a soil. If we take all biocenosis out, all the microorganisms, all the animals, everything, then of course soil is dying. And then when it's dead, of course, it regenerates very, very, very slowly. Let's look at organic matter, what we just heard. So what kind of organic matter is in soil? First, it's a living organic matter. It's all the microorganisms. Here in the middle of picture, I'm sure there are a lot of them, plenty. Different worms and bugs and bacteria and fungi and such. And of course, then there are bigger animals like rats. And since I couldn't find in my archive any picture of rats, and my house cats will do the picture of bigger animals which live in the soil and gives a lot to the soil because they burrow it, they turn it, and it makes soil different when there are these animals. When they finish their life, they of course become a debris of organic matter, which is basically a lot of plants, but of course there could be animals, and of course dead microorganisms too is a debris of soil. And quite huge and important part of soil is different kind of, let's say, poo. Here it's a cow doing, but of course bacteria and fungi and every living organisms gives out some matter that is already changed, and it changes environment and gives part of all the matter to soil. And then when microorganisms have worked around all the debris, it ends in humus. And humus is this beautiful thing in soil, which we can see in the upper part of soil, which we can call a horizon. And it gives us this blackish color on our fingers when we touch it. And of course, depending on soil, depending on where the soil is, it could be very rich, like here, and then almost non-existent, like in these cases. Why it is so important that we speak about organic matter, why organic matter is in all our thoughts and on our tongues. First of all, of course, we did all a lot of bad things with dead dinosaurs that we put in our cars. So we need to think how to put back carbon back in soil. And soil actually can contain quite a large amount of carbon, about 5%, but if we manage to put it deeper, then it could be more, of course. Then the next thing is that organic matter is very, very hydrophilic. That means that soil, which is rich in organic carbon, could contain much larger amount of water than soil without organic carbon. And of course, this is more important in places where it's colder climate. And in the springs, if soil is dark and rich in humus, in organic matter, it is quite easier to warm up. And since it's moist and warm, the capacity of soil to store the heat and in the same time not to be overheated and bring this very fluent flow of heat to and warmth to all the roots and living organisms, it's very, very important. And it is very important to farmers who rely on early vegetables, for instance. Early vegetables usually are expensive. So every farmer wants to get more money. And if they have this possibility to get like maybe 1 week or even few days of their vegetables earlier to the market, that means a lot of them. And only if the soil is capable of this miracle to bring the worms to soil earlier than in other soils, it can happen. And of course, soil organic matter is storehouse of nutrients for all the biocenosis. It starts with bacteria, which break down organic matter, releases minerals, which is then goes to plants, and plants, of course, goes to herbivores, and then, of course, there's omnivores and predators who eats it all. So if organic matter is rich, then there is a possibility to keep up biocenosis. And then, of course, organic matter reduces soil bulk density, which is very important if there is heavy texture in soil. And of course, it helps bring soil structure. Why soil structure is so important? For plants, not only, and of course for microorganisms too, and for small animals, it is very important not only moisture in soil, but air too, oxygen. So if there is very, very dense soil with no micropores and bigger pores, there is no possibility to normal growth of plants and such things. So it is very, very important. And soil structure depends on organic matter and on soil texture, of course. Is it clay? Is it sand? Is it silt? And then, of course, some ferrum and or iron and carbonites could be involved too in more heavier places. So when we look on the soil structure here, I took a picture of, it's like a 0.5 millimeter of soil structure. What we see here is a little pieces of silt, small pieces. And then this darker matter is organic matter, which keeps it all together. And imagine that it's like not even a pinhead. It's a 0.5 millimeters. We see this beautiful, magnificent structure that keeps it together. And one fact that I didn't put it down, but imagine in 1 gram of soil, you can get from 80 to 800 square meters if we make it flat. And this is absorption capacity where water and nutrients and everything can be absorbed and stored. And of course, soil is a beautiful system that comes together in when atmosphere, hydrosphere, lithosphere, and biosphere works together. And this is a beautiful cross point of all these spheres on our planet. What factors make soil? So first of all, of course, parent material. This is what, or lithosphere. This is what maybe glacier, maybe some volcano, maybe sea brought us. So it is maybe clay or silt or sand or volcanic material or something. What is nature has give us. Then, of course, relief. Again, nature. Then, of course, time, because this is a result of processes. So every physical and chemical process needs time. Then, of course, climate and living organisms, what we talk so much. And then, of course, there is anthropogenic factor, because we as humans can be count as living organisms who, like every other, eats and poos and then dies. That is one way of our life. And then second way is, of course, anthropogenic factor. We influence a lot on our planet with our doing. So let's talk about what we can do with soil. First of all, how we can influence parent material. Basically, we can physically move it somewhere. We can remove, we can add, we can stop, like terracing and such things. And then we can, of course, change in this way our soil, let's say, make sandier or make more clay. But usually, if we want to make better soil, it's, of course, takes a lot of money to do such thing. Then, of course, relief can be changed quite easily, actually. Terracing, of course, is very important if the relief is very steep. And leveling happens a lot if the relief is like with small waves, and we want to make them very level to use it easier with big machines and such thing. Time, thanks God, we can't make any impact on time. I'm sure that we could do something wrong, like we did with climate. And climate, of course, we can change globally, and we already did it, not on the best way. And we can, of course, make some changes locally. And this is usually happens when agriculture is very intense. Then we water, then we drain. We can add temperature. If it's in some greenhouses, we can add light, change length of the day and such things. And of course, then plants and everything grows better. And then we can influence, of course, living organisms. Of course, we kill them because we want to eat. But now let's talk about what we can do to not kill them more than we need. And I like this picture with petunia so much. I took it one day. This black stuff here, this is not soil. This is asphaltus, concrete asphaltus. So the petunia is in a small amount of sand, maybe some spoon or something like that, and it lives. And nature really is so resilient against us. So best ways to kill soil, how we can do it easily. First, we can cover it with concrete, asphaltus and everything. And of course, if we want to make our living better, we need to let in our cities as much as possible places without cover. This is in our best interests. Then, of course, it is very a good way to seal the bogs. So I even last summer went to one conference where there was scientific paper about how good it is to seal soil, to seal bogs with mineral soil, because then we can store all the organic carbon, and it is not going up in the air, and so we do good. But to think that all the biocenosis, what was there in this peatland, is killed instantly by sealing it. And of course, the capacity of this peatland to take in water and store it and then give it back to nature, to the rivers and everything, is already shut down. It is not possible anymore. And we can, of course, only cry after what can happen if the big floods come and there is no peatland which can store this big flood water. Of course, overgrazing is very easy way to ruin soil, particularly in places where plants grow slower. For instance, here in this darker picture, it is from Iceland, and there is enough water for grass to grow, but there is not enough temperature for grass to grow quickly back. And here this is in Mediterranean. This is, of course, enough temperature for grass to grow, but not enough water for grass to grow. And when we overgraze it, then again, it's a problem. Low mowing is always often very, let's say, huge problem. This is from Latvia, and usually the grass in Latvia grows very easily. But then when we cut it very low, we ruin the center of plant, and then comes 1 or 2 weeks or even 3 weeks of heat, and everything is killed. So it is easy to ruin even very, very good growing grass. Then it is easy to erode places where soil texture is light, like here, it is loamy sand. And if the terrain is quite steep, of course, we can see that here soil in the upper part of leaf is without humus and almost no organic matter existent. Fire always is bad for soil. Of course, we can discuss that fire brings some beautiful biocenosis with rare plants and such thing. But when we speak about soil, it's always bad. There is no good way for soil when there is fire. Excessive tillage is a problem often when we try to store moisture in soil, as it in this case. Here, 6-7 times a year, tillage is happening only to store. And if we imagine that it is clay, heavy clay, which is brought to this dusty stage when it is not even, let's say, keeping straight wall in the pit, it is very bad way. No structure. And when we do this, we go in this continuous evil loop. We tillage because we want to soil stay moist, and since we tillage, microorganisms is easy working with air, with oxygen, and breaking down organic matter. No organic matter, no water, because there's nothing more to keep this water. So this always ends bad. And then, of course, the worst, the best way to kill is bactericides. It is actual case from Sicilia, where in 70s were bactericides used in soil to kill soil-burning diseases, and it is still not got back in good state, let's say. There is no structure. We can see this here clearly. What we can do when we kill soil, how we can put it back. So first of all, microorganisms, bacteria, all the centropods, all the earthworms, everybody needs to eat something. So we need to put back some debris of organic matter. That means straw, grass, organic manure, everything that we can find. And of course, the more variety to diet, the more plants, the more different microbiome, because every plant takes with it different microorganisms. And then, of course, main microorganisms which is good for soil, like nitrogen fixating and such, needs a pH around 7, which is usually not very hard to get in Iberian Peninsula. But here, these both pictures are from that place. This is around Sevilla, and it is like almost pure calcium carbonate. And this is around Evora in Portugal. And this is mainly silicium oxide, which is quartz. And it is very low pH. And so it could look similar, but this is two completely different cases. And of course, water. Microbiome needs water because bacteria, of course, can't grow if there is no water in the media. And here, depending on how much water is in soil, we can see how this humus horizon grows. Here, very, very dry soil. And then there is more water and more, and it goes darker, of course. And we can see, of course, how lower parts of soil changes too. And then, of course, is the main rule: you need to give more to the soil than you take. Often, sometimes, let's say, organic matter is magic thing, but not to that place where you can't get from organic matter what you do not have in your parent material. In some soils, there is no potassium or no phosphorus. From thin air, we can bring in soil only nitrogen. some ash minerals we need to bring in. So we need to consider that bag with mineral fertilization is not always bad. If there is nothing in soil, we need to take it somewhere and put it in soil. Okay, and then last two pictures from my country. Both of these are sand, pure sand. The picture on the left is from the sea coast, where we can see how coastal erosion kills soil, and we can see how organic matter flows in the sea. And this is a picture from farm from one of my students who works quite intensely on soil. He grows wheat, and this is sand with beautifully made organic layer. Even in very intense agriculture, you can give back to soil and keep it alive for long periods. And that is all from me. Thank you.
Thank you so much. You have a beautiful presentation. Really love your enthusiasm and being a soil ambassador. It's really, really good. We are slightly over time. Yeah, I had a question, but if you mind, I will take this question at the end because you just
Of
touched
course.
this example of wheat, and we now have a line. The Rocio. Rocio is going to tell us exactly about wheat and microbiome solutions for soil restoration. So, Rocio, I call you to the floor, and I also ask our audience if you have questions for Ieva. I will take note of your questions and raise them at the end. Okay, so please use the chat because I'm always with an eye on the chat. I can always take them later. Rocio, all good from your ends? Okay.
Yes. Let's see if
Yes,
you can see the presentation. Okay, 1 minute.
we can see, not in presentation mode, but it's here
It's-
up in the screen.
Okay, it's coming.
Again, we
Yeah,
have the notes view. Okay, so
don't
you can
worry,
just
it's fine. Now
perfect.
I fix it.
It worked
Yeah,
very well this
okay,
time. Thank you, Rocio.
perfect.
Go
Yeah.
ahead.
Hello, first of all, thank you very much for inviting me for this session. I'm very happy to participate in this Food for Sustainability Academy. Well, I am Rocío Barros. I am a researcher at the University of Burgos, and we work mainly in soil, not only in the agricultural soil, also in industrial soils, and in order to see how the microbiomes can support on improving the soil property, but also the remediation of the soils when the soil is polluted. But today I'm going to present the soil microbiome in the Mediterranean with cropping systems. That is this part of the work is mainly developed in the Tribiome project. It's an European project where we are studying how the microbiome of the wheat is related also with the food that we are eating, but also with our intestinal microbiome. So we are working on in order to understand how all this is mainly related. So, well, just to give you an overview a little bit of the project. So we are different partners, and what is mainly interesting is that we have different partners and different fields across Europe, but also we have agricultural associations, and we have different companies like Fertility, for example, that is also developing some of the strategies, sustainable strategies based on microbes. So just to explain you a little bit why this is important, and then later we can also see some insights about this result, is that well, we have selected for this project wheat. So we selected two types of wheat, Triticum astibum and Triticum durum, because it's one, well, Triticum astibum is one of the widest crop used in Europe, and we wanted also to include a different wheat that is also, the production is also increasing in Italy, France, Spain, Greece, like is the Triticum durum. So the main idea is that we can study the soil, I mean the soil, the microbiome of the soil and the microbiome associated to the wheat production, and we can understand how this microbiome is affecting the quality of the wheat, the quality of the flour, and also how the product developed from this wheat are having an effect in the gut microbiome, not only in humans but also in different animals like cows. And the idea is also to identify different biostimulants, mainly based with microbes that can change or in order to see how is the effect of these microbes over the global microbiomes from the soils, from the rhizosphere to the gut microbiome in the cows and in the human gut microbiome. So we set up a methodology, we select the different fields, we sample the fields, and also we select the different microbials and non-microbials modulators in order to see this effect. And also we develop field implementation in order to get new insights and good results for this research. So we are currently evaluating different fields in the Mediterranean area. So we have different fields in the north and in the south of Spain, and two fields also in Italy, in the north and in the south of Italy. But also we are evaluating fields in South Africa. But today the results that I will present are more related with the European part. So we were for this, it is very important also in order to understand the microbiome of the soil. It is very important to define when we have to sample the rhizosphere of the soil. So we develop, and also how to sample this. So we develop a protocol in order to sample this rhizosphere, mainly related with the wheat cycle. And yeah, we just develop a common framework in order to be able to analyze this microbiome from the different sites. What it is important is that, well, as you know, the European Commission is also developing, well, we already approved the directive for soil health monitoring and the soil health monitoring law. And the thing is, like, of course, we try to follow also the rules that has been established in order to that this data that we are also generated can be used for all this data and all this research that the European Commission is doing in our analyzing the whole microbiome from the European panel. So yes, we took different type of samples. So we took root samples. So in total, we were analyzing 1,400 roots. Also soil samples. We analyzed 198 soils, and also we were taking some plant samples in order to see the quality of the plant. And for all this, just of course, this is a complex, but just to let you know that for all this, the idea is also that we are able to analyze which is the microbial community, but not only the microbial community. I mean, not only who is on the soil, also to understand how they are performing the work, and also to understand which is the function, why these microbes are there, and which is the metabolic route, or what they are doing in order to increase the quality of the soil. So, well, just as a summary for this, we were also analyzing bacteria and also fungi, and in order to identify who are the main drivers or who are the main microbes in these soils, but also to understand which are the functionality. And just these are some pictures of the different campaigns that we were performing, and yeah, so we were just taking different samples from the roots and from the soil. So just to present you a little bit some results, just to let you know that we were evaluating the different soils, and we have seen that, of course, these soils are completely different. Mainly, the one that has a very, very different situation is the one related in the north of Spain. So the one that we took here in Burgos, mainly because these soils have, as I think it is good because it's related what it was presented before, is related with the high organic carbon content. So this soil shows like a higher nutrients, higher organic matter, and that makes the difference from the comparing to the soil from Italy and the south of Spain. So if we see also the enzymatic activities, I mean, what is going on in the soil related with the microorganisms. Also, what we were saying before is what we can see here is just that the soils from the north of Spain show a higher enzymatic activities and mainly related with the carbon degradation. So, I mean, that is it makes sense because we have this with these soils with high carbon content, so the activity of the soil present a higher enzymatic activity related with carbon degradation, that is the beta glucosidase. Also, we see that in the other soil, the enzymatic activities is lower. Also, that is also related, a part of the higher carbon content is because the humidity and the water content of the soil is higher in the soils of the north of Spain, while in the south of Spain, like Sevilla, Cuenca, and also in Italy, in the Tuscany or Sicily, the soils has lower water. Regarding the microbial community, the question that we did, or the question that we wanted to answer was if there is a difference between the microbiome in the soil when we are in normal or in a high aridity conditions. I mean, if it is rained or it is a rain area or it is aridity conditions, and we wanted to know if there is difference between the microbiome of the soil and also with the microbiome associated to the root of the crops. What we have seen is that the pattern, we follow the same pattern in all the cases, and is that when we are analyzing when we analyze only the microbiome of the soils, we cannot see difference when we have a normal on a high aridity conditions. But however, we can see difference. We can see that the microbiome from the roots part from the rhizosphere is completely different when we are talking about the dry or wet areas. It happened with the Triticum astibum, but we follow the same with the Triticum durum. So we see that the microbiome associated to the rhizosphere is different when we are in a wet and a dry, but the soil microbiome, it doesn't present a significant differences. And also we see the same in Italy and Italy with the Triticum astibum and the Triticum durum. So I don't want to go in detail, just to let you know, and the key message of this is just that the wet and the aridity conditions can influence the microbiome that is associated to a crop. And just we were analyzing also the different microbes that were associated to the different rhizosphere, and we just got that there are some phylum that are common in the different from the different crops, from the different soils. And we saw that we have like proteobacteria, rhizobium, pseudomonas that are involved in the biochemical processes, but also we have actinobariota that are related with the degradation and transformation of the organic matter, which is which is related with the fertility of the soil. So understanding who are these microbes, I mean, which microbes we have in a soil, we can understand also the health of the soils and understanding which are the processes that the main processors or the main drivers that we have in this soil. So yes, some conclusions of this is that, well, to have this methodology allowed to compare the data, and as I said, the resource per microbiome shows significant differences, and we have common phenom in the soil and in the resource per samples that support in the processes that we have in the soil. Of course, this research needs to continue because it is important to understand or to find common microbial species for the different crops, from regions, from the different treatments, or identify which are the function of this microbiome in order to understand how to modulate and how to increase the health of this soil. Just to explain you a little bit about other issues that we are doing in this work is a part of our study, the microbiome of the soil. The main idea is also that we are able to isolate microbes from these sites that that can be applied as a biostimulant. So, for example, we isolated 100 microorganisms, but we have selected two modulators. One is a bacillus and one is a pseudomonas. And we were studying how this microbe can support the plant in nitrogen fixation, different hormones production, solubilization of phosphates. So we were analyzing. And we were validating these modulators in different field trials in Italy and in Spain. So the same where we were taking the samples. And we validate this application of this biostimulant in the background fertilization and in the top dressing fertilization of for wheat. And we were also trying to see if there is an effect of the modulator when we are reducing the fertilization, and we were also doing different combinations. So as a main result, so just to give you an overview, is that we have seen that Pseudomonas works better as a biostimulant. Also, when it is applied as a liquid fertilizer, as it is expected, this is working better in the rain-fed condition than in irrigated condition, because in the south of Spain, so in some cases wheat is irrigated. Also, under irrigated conditions, what works better is the combination or the mobilization of the bacteria in a fertilizer. And in terms of application, what it works better, it was application during the top dressing. So yes, as a summary of I was explaining today. So we were trying, I mean, of course, this is a preliminary research and we are continue, but our main goal is in order to, I mean, at the end of this year, is that we are able to identify the key drivers for the wheat species, considering irrigation and non-irrigation, the region, and also the temporal evolution of the crop, and also to see and to be able to get the real insights and the real information of the soil microbiome, the structure and the function. So key findings that we already have is that the soil properties drives the microbial biomass and the enzyme activities. Also, during summer, we have seen that the microbial function is decreased, of course. Also, we know that the carbon use of the different microbes shifts from simple to a complex compounds when we are changing when we depending on the region, and also we are also analyzing right now, and we know that the microbial structure reflects the plant nitrogen level. So also the microbes that we have applied change the nitrogen level at the plant level. So yes, these are also some of the pictures of this of all this work and yeah on field. And yeah, thank you for your attention. This work has been also developed in collaboration with the University of Bologna. And yeah, and thank
so
you very
much,
much.
Rocio. It was a pleasure to listen to you. Great presentation. While I call Ademir on stage, just a clarification question. You mentioned at the beginning that you were testing the quality of wheat. Did you test for things like the nutrient profiling of the wheat, or you're more looking at, plant growth and root growth and other kinds of in terms of the plant and the culture?
Um,
Hello, Rocio?
She was with low
I
battery.
think
I think we lost.
Yeah, I think we lost Rocio, but we'll continue the conversation when Rocio comes back. And we also have a question in the chat. Thank for this. So, with no further ado, I think we can call Ademir. Hello, Ademir. Cannot see you in the screen, but I guess you're there. So we move from very, very cold countries to very warm countries. So now we go into the South Hemisphere. We go to Brazil. Ademir, can someone help me to see where Ademir is?
Yes, right.
Ah,
Okay, good afternoon.
yeah. Hello, good afternoon.
Hello,
How are
good
you?
afternoon, everybody. I'm fine, and all of you?
Yes, doing well.
Great.
Thank you so much. So, Ademir, I don't
Thank
know
you.
if you have a presentation
Yes.
for us. So if you want to share your screen, and
Yeah.
also
Fine.
thank you for being here. I think it's
Now,
early
here.
hours in Brazil, right?
Just to check here. Okay. Ali, mas cadê
Okay.
onde é que tá?
Não, eu acho que
You have the
Onde?
share or compartilhar.
Não é, Mari.
pede para ela ajuda, compartilhar.
Can you help me here? Pode me ajudar
Yeah.
aqui?
Compartilhar ecrã. Tem que compartilhar.
Compartilhar aqui, sim. Tela.
Pronto, e as telas, deve encontrar aí na
Já
tela.
vou direto aqui na apresentação, né?
Sim, é isso. Já deve dar. Assim que nós virmos deste lado, eu aviso. It's coming. Okay. It's coming.
Ok?
Yes, perfect. Right on spot. Okay. So we're ready to go, Ademir. Okay.
Onde é que tem que pressionar aqui agora para ficar inteiro? Como faz?
It's perfect from this end.
Tá ok?
Yes. All good.
Okay. All right. So let's go. Okay.
Estamos a ver a
Just have
posição
a
do
look.
Brasil.
Here is Brazil. It's quite a big country. We are here in the South Brazil, in Paraná State. It's one of the most important agricultural state in Brazil, in all 27 states. We are responsible for a quarter part, more or less 23% of the grain production in the country. And so we discuss a little bit more about the soil management, how we are achieving in regenerative agriculture. We have a lot of estimative, some is in Portuguese. You can see here the FAO data show that more or less a third part of the world, the soils are in degradation process. And also, if not to be different, if you not take another way, we achieve more than 90% of the soil in 2050 with the problem with the degradation. Okay. So this is common practice in 70s in Brazil. was the soil plowing, or say, the disturbance of the soil and the big problems with the water erosion, the seeds, the fertilizer, everything. And also loss of water, soil organic carbon, nutrients, and many other particles. So what the main reasons for the use of cover crop service plant? How you make a better management to achieve these good results? One thing is looking for this conservation agriculture system, no? Is looking for this system and to put together the minimum soil disturbance, the biodiversity, not plow the soil, and to remain, if it's possible, the soil covering all the time in order to achieve the good results. This equilibrium of the different systems, the integration of the system, the soil management in a sustainable way. So use of cover crops with quality mix, preferentially adding or equilibrium of the nutrients in the soil, organic or some mineral that you need, and also soil pH correction or some aluminum with the use of lime or gypsum on others, and also the balancing of the micronutrients, very important, and also the improvement of the bio insumos, ou seja, biodiversity with different bioactivities, bioactivators, also composting. Sometimes we use a lot, we have maybe 12 million hectares in Brazil, integration, grains and livestock, and also adding different sources of carbon. Look for the equilibrium of the all attributes, soil attributes, physical, biological, and chemical, and use the rotation, or say, diversify different crops, diversify the different system according to the region, according to what the farm wants, and also the all agroecological conditions, and also to promote microbial stimulation, and reduce the use of pesticides. So in Brazil, we know in the science that we have quite 160 more is 1,000 species of fungi and bacteria that living in the soil. The total number of species is just that more around 12% of the diversity. So we need to develop technology, biostimulations, to improve the biological activity of the soil. This will be very, very important to achieve this. So just have a look here, the situation of Brazil. And Brazil has one of the most largest area of the adoption, 20% of the global farmers adopting with the biologicals. Brazil is leading. You can see here biocontrol adoption here. You take this to see later on, you see 55% currently used. You see here biostimulation. You can see here biofertilizers comparing with European Union, USA, and many other countries. So Brazil is leading this aspect. We'll have many problems, many challenges, but how to use it quite a lot. And I will show you quickly some farmers, some results that some farmers are achieving here in Brazil. We are working a lot of the research. So I did my master in Aberdeen, Scotland, 94, 95, and did a PhD here, and also in Kansas State University in Manhattan, and also in Université de Paris, together with the INRA, Institut National de Recherche Agronomique. And here are some farmers that we are working with them quite many years, 6-7 years. This is a farmer with 500 hectares that use this all aspect together: cover crops, biological, equilibrium, no-till, diversifies. They grow soybeans, they grow maize, they grow sorghum, they grow bean, they grow wheat, and also some different species, mix of mixed cover crops. You can see here the results of the soybean. You can see here the knife roller with the planter over the falling down the mixed cover crops, and the soybean growing this. And you can see the high yield that they are achieving. This is started with the 60 bags. This is mean 3.6 tons until 92 bags. So 5.5 bags in few years. What's the response for this? Not nutrients, not inputs, is not biological. Is biological, is cover crops, is equilibrium, is no-till, is micro biology, biodiversity, a lot of fungi, equilibrium on the soil, more also increasing soil organic matter. This farm, Lely's family, next week we have a very big meeting there, big field day, maybe 400 or more farmers and technicians, agronomists, the different states coming, because they are developed quite strong regenerative agriculture in this area. Is very clay soil, more than 60% of clay, no compaction, no nematodes, phytonematodes, no soil disease, very, very low, because they use this equilibrium. They decrease 35% the total costs, total farm costs, crop costs, and also they decrease 65% of herbicide. In 3-4 years without applying potassium, because they use a lot of different mix. For example, finger millet, that's the most efficient to cycling potassium, that go 2-3 meters to up these nutrients on the top. And also some radish and some other species that look for the sulfur. They use a lot of legume as well, or say different species, some that also can promote the phosphorus solubilization, achieving this. We have some other farmers, this in Minas Gerais. This is a farmer with 1,700 hectares. In some Okada, they use integrate with animal. They use here oat, white oat, black oat, rye, radish, buckwheat, common vetch. We decide together with them. They use animals. They also they plant maize, 16, 18 tons per hectare. They use beans. They use wheat. They use soybean. It's quite strong. They make composting. They use bioactivators. They use different cover crops, different biological, pseudomonas, bacillus, different kind of bacillus, subtilis, aminoliquefacients, and many others. They use azospirillum, trichoderma. They use mycorrhiza, different biological products, actives with the strong results. You can see here some other farmers using common vetch, hairy vetch, black oat, white oat, rye, and red together, different species, different families. They use overgrazing to regrow and plant maize, soybean. So very common this in Brazil, decrease herbicide, improve nutrients, improve soil life, earthworm, microorganisms, so achieving the strong equilibrium. You can see here, for example, this is from the left to right. This is buckwheat. This is Cajanus cajan. Oh, sorry. This is buckwheat. Cajanus cajan, radish, frutillaria, some are legumes, the tropical legumes, Juncia, sunhemp, crumby, abyssinica, and also sunflower. So different species, different effect on the soil, more enzymes, more different root exudates that can promote more biodiversity in microorganisms. You can see here these friends in Savannah, in Cerrado from Brazil, our friends, very strong people. This farm is 2,200 hectares. They use maize, soybean, sorghum, sunflower together with mixed cover crops, sorghum together, intercropped with mixed cover crops. They use rice, beans, they roast also cows, integrate all of this. Is a circular fish, is a wonderful condition, regenerative, strong yields, and less costs. So we're looking always to living soil, to promote living soil in different conditions. This is a long-term experiment that we have from our IEPAR. I'm retired for 14 years, but I continue research visitor. This experiment has now 40 years, exactly. University of Germany, University. We will be here, Dr. Maria Fink, that we work together in this experiment. They will come in September. Next September, we will celebrate 40 years. We have here 14 theses, master, PhD, post-PhD, 34 different articles, scientific articles, studying 0.2 to 5, 10, 2 meters. The soil have 37,000 soil samples during these 40 years. I started this in 86 with our friends, and these different cover crops rotate with maize and soybean. Results here in soil, organic matter, and organic phosphorus, microbiology. My thesis of master in Aberdeen, 94, 95, Scotland, my PhD, also Brazil and Kansas was here with the carbon sequestration, 2006, 19 years ago, and many other theses from Brazil and other countries. So trying to study to understand this system in southwest of Brazil. And here, a lot of papers showing that when you put different species together, when you promote the biodiversity, the rotation, you can see here the soil organic will go into the direction of the forest. So the main, when you have no-till, was 19 years, now it's 40 years. You are in direction of the forest, you are in direction of the mother nature with the equilibrium, biological, physical, chemical, and with more soil organic carbon retention. You can see these, they use a lot of this to manage our cover crops or crop residue. The knife roller, very common here in Brazil. You can see the plant, they go very quickly because I have a lot of slides to show you. This is a mix of 7 different cover crops, 49 days. We are just past knife roller and planting. It was perfect. And also this is for soybean, for maize, for cotton, for wheat, for vegetable. You hear our friends, ladies group here in Brazil. This is a coffee, very famous coffee. This coffee was served when the Prince Charles in England became the king. I did my master. I have a friend, Stephen Briggs. He's a very famous man. He works for 24 years in organic with the 6 or 10,000 hectares now with the royal family. And this coffee, Monserrado was selected among 42 different coffees. It was the best one. It was served for more than 2,000 people, more than in around 200 countries in England. So this is a coffee of this lady and his family in his farm using this regenerative coffee, the high quality. Here is some Japanese also using this mix, this approach of this regenerative agriculture. In Brazil, this was the largest Sequita family with the carrot productions. They have more than 4,000 hectares, the whole family, and also different potato, tomatoes, all of this, carrots and others, to produce in this sustainable way. You can see here the soy after mix with the biological activities and bioactivation. The difference decreases the Brachylanchus brachyurum, the main phytonematodes here in Brazil, the lesion nematodes. We have a big problem here in Brazil. The 3 big problems in agriculture is soil compaction, is root disease, and phytonematodes. This is the big problem in Brazil. And also you can see here 25 bags. This means 1,500 hectares, 1,500 kilos of soybean, more when you compare the system, when you use the cover crops, the biological compared with the pattern of the farmers. So when you use this diagnostic adequate in different equilibrated areas, homogeneous areas, you can achieve good results. This is very important point. You can see here what we're achieving in nutrients aspect, more than 130 kilos of nitrogen with the mixed cover crops, phosphorus, more than 380 potassium that goes down because the plants, it's a lot of result international, the plants was not passed by the engineering development or mejoramento genético, que não passaram por esto, that not through this, they have like a forest, they go 2-3 meters to uptake nutrients, not in the case of soybean, maize, wheat, and other plants that was studied. Então, this is very important to use this. For this, you can see here the different species that are using together with maize, with sorghum. You see reddish oat, different oat, white, rye, triticale, and many others that use here. Here, a farmer of 7,000 hectares, the Netherlands people here in Brazil, they use together with pasture harvest after maize and after come to graze by cows. So this is very common here in Brazil. You can see here also in Canada that will be there with Dr. Odette Menard, is a very famous lady working with this approach. You can see here the results of the radish. You can see the effect of the radish with the acid phosphatase, the enzyme from the roots. 98.5% of a microorganism, they out-solubilize phosphorus. We have a different mechanism, citric acid from the lupin. We have the different plants that are host of the mycorrhiza. This is also psicetic acid, also for PGMP, that's important to solubilize phosphorus in different systems. You can see here, all biological fungicide, insecticide, nematicide, very common. You are increasing this, decreasing the costs, improving the soil life. Soil life, living soil vivant, we will increase the soil capacity or productive capacity, and also for different mixed cover crops. A different quantity, amount of nutrients available for these different mix. I worked these last 27 years with the different mix. I developed different mix, and I work with Rolf since 1977, 49 years with cover crops. I've been working with 172 different species, variety of cover crops, and I did more than 135 different mix for different special situation, 45 days, 60 days for forage, for soil, for different plants, for different conditions. Different results that you're achieving the yield with the maize. Here is Ray Archuleta. I talked yesterday with him. This is one of the Gabe Brown rays are in USA, very, very famous in the world. One of the most famous in the regenerative agriculture. Our friend here visiting Brazil. This is Alejandro Carrillo, also very famous in agriculture in Mexico and other parts of the world, in Australia and others, very poor, very less than 200 millimeters in soil, in totally almost without rainfall in desertic areas. And here my friend Frederic Thomas, I received a message from him today. He's a friend of us. We met the first time in 2001 in Spain, in Madrid. This is a big the first CA World Congress. We exchange a lot of information. They use a lot of these mélanges de plantes de couverture in France. He's really a fantastic. He developed this knife roller in front of his tractor, and he's with the lamb. And here my friend Sergio Argenteri also that are doing very nice work in close to Genova and in Italy. A lot of utilizing 16 or more different species, and are doing maize, wheat, and other crops perfectly. This system, when they not deal with quality, with the regenerative agriculture. This is our friend Gabe Brown, USA, fantastic man that did a revolution in USA. Here in Dnipropetrovsk, just show you that it is possible to work in any part. This 10, 12 below zero in Dnipropetrovsk in Ukraine. I've been there 6 times, in Russia and many other countries. So this it can work according to species. This is in Portugal. We've been working with our friends in Portugal, learn a lot with André, with Paulo, João Valente, many others that are doing very nice regenerative work in Portugal. This also in Samara, Russia, yellow sweet clover. Is fantastic. This is a man very famous, Franz Jetman from South Africa, working very nice with mixed cover crops. He developed this knife roller for grapes and many other different systems. This in Ukraine, this knife roller, we put different mix here. You go to the end now. And this is in Africa also with the Crotularia. You mix with sunflower here for 400 millimeters. In Eritrea, I have 44 different dishes in Africa, different conditions. In Mozambique. So, following Tuesday, I will finish. Feed the soil, not the crop. And the soil will feed the crop. In cover crops, is biology primary enhanced no-till soil performance ecosystem system, like a bioactivation, or say the domino effect. Biology, chemistry, physical, soil health together is the only form that you can change, exchange, you can move your soil, all attributes, chemical, physical, biological, with the plants, with different species, biodiversity, like the previous speakers said about this importance of microorganisms. You are working very strong, our farmers also, about that. And you can see here the regenerative agriculture together to achieve more productivity, more profitability, and sustainability together, of course. and also
More
the system
resilience.
with more resilience. We go to the end. And a sustainable agriculture disequilibrium with cover crops, if you can integrate with animal, with trees, with grains, the no-till, with the rotation, quality of the farmers, and also soil and plant bioactivation. Soil healthy, plant healthy, food healthy, and people healthy. So healthy soil, continue root system, growing green breach, minimize soil disturbance, maximize soil cultivation, maximize biodiversity. And regenerative sustainable agriculture system must comprise biodiversity, soil life, and many other aspects. And we need urgently bioactivate our cropping system, recover the memory of productive soils, harmony, and abundance. Sorry for the time.
That's brilliant. Thank you so much. Thank you for the energy and thank you for such an empowering presentation. It was very inspiring to hear. I know that we are reaching the end of this session, but before you go, and while I open for also for a couple of questions with our speakers, I will just kindly ask our team to share our final quiz. As we mentioned, we're going to have some questions for you. You can, fill in the questions while we open now the discussion with our speakers. So Sophia, thank you so much so you have here the QR code. Also, eventually we can have a link in the chat if possible. And please do feel free, take your time. And now I will just kindly open the question. So we started by talking about biocenosis and its importance. We also talked about the soil microbiome and the soil analysis. And now we've been through, how cover crops can stimulate this soil microbiology. So I will start with a question for Ieva, but maybe the Rocio and Ademir, you can also take your perspective. So beyond this basic chemistry, so we are talking about the NPK and the pH. Which accessible biocenosis indicators, like, for example, enzymatic activities, genetic sequencing, microbial biomass ratios, but so which biocenosis indicators do you think farmers should track to assess soil health trends over season? So what are the indicators that can lead us to an understanding if we are actually going the right direction or not? So feel free to...
If I need to answer this question, the soil, of course, is the thing that do not change just like this. So you can't get very clear answers in one summer.
But if you were measuring if these interactions between the organisms, the plants, the animals, so all the interacting organisms that then form these interdependent relationships that you so well explained, do you suggest any specific measure to track this?
In case with microorganisms, you can always measure intensity of, let's say, breathing. When soil is alive, it breathes. And you can, of course, this analysis is quite old, quite known in the world. So you can easily say if there is anything live in soil, and you can check it, of course.
Okay, and so from your perspective, Ademir.
We have Dr. Ieda Mendes, the very famous scientist lady. Next week we'll be together in this field day. This lady and a group in Cerrado, Savannah, in Brapa, they developed almost around 20 years. I studied very interesting. And it was published in English in USA. It was the first one in the world. They are very, very interesting. What they did, they put a soil, uncovered soil, and comparing with soybean, with maize for many years, and tried to, and also with the cover crops, mainly brachiaria. And during these years, They check what's happening with soil organic matter, what's happening with the organism. They saw the more organisms with the cover crops. They saw that there were more crop yield of the soybean during the years. They saw the increased soil organic matter. They saw the increase a little bit more the nutrients. But when they check if it was the nutrient, which nutrient, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, no, there is no correlation, very low correlation. When they check the organic matter alone, no correlation. They saw micronutrients, no correlation. They saw what it did. They evaluated the enzyme, and the enzyme was fantastic. So they did 12, 13 different soy enzyme, soy plant root enzyme. But now they are using, Brazil is mainly 3 that are real sulfatase, the beta-glycosidase, beta-glucosidase, beta-glucosidase, and mycorrhiza, and this evaluation, the microorganism. So they saw that, for example, there is a strong correlation. When you have more this enzyme, what this result is quickly saying, very in short words, as more beta-glycosidase do you have in a plant tissue, more this residue goes in the direction to form to become humus and less lose the atmosphere. But how you can achieve more beta-glycosidase? One of the main aspects is more biodiversity. When you put more biodiversity of plants, when you put more biodiversity of microorganisms, more beta-glycosidase. It has more beta-glycosidase in the residue, crop residue. They go mineralize. When you go to the end, they have 2 ways. One go to atmosphere, one go to direction of humus is more. So this was very, very interesting. So we have the 3 big bioindicators: soil organic carbon, soil organic matter, mycorrhiza. Mycorrhiza is very difficult in Brazil because the temperature is very high. You need to immediately in good condition, good temperature, and go to the lab to evaluate. But earthworm, soil organic matter, and soil organic carbon, and mycorrhiza.
Okay, and you also mentioned a bit of the enzymatic activities
Are you
to measure. So on that matter, you mentioned a lot of cover crops and a mixture of cover crops. My question is, how do you choose that mixture? So what preliminary analysis do you do to select a specific cover crop mixture?
Yeah, we have a very strong evaluation, because we need to know with the farmers with the main aims, and also the soil. We need to talk with the soil, talk with the roots, the calicato, the trench, and see what's this plant need, what they want, what is the main constraint, what the main challenge there. If the less water, we look for some species that can survive, can support, that can go. So there is some soil compaction. We have many different species. We need nitrogen. You have species very efficient, 200 kilos. We needed to look for bring potassium, because potassium in sandy soil goes down. We saw the last year here in Goiás, in sandy soil, during a week, 180 millimeters of water, the potassium goes to 60 centimeters. We know that the annual crops, the great majority, is 20, 25 centimeters, 88, 90% of nutrients are there. I work a lot of coffee. With the coffee, it's 4 or 5 meters. So 40, 50 centimeters of the roots on the top, up layer, 90% of nutrients are here. If you go down 2, 3, 4 meters to look for water, 60 centimeters in soybean, go 3% of nutrients. So we need to save the nutrients, improve all this micro, meso, macrofauna and flora, biodiversity, different root exudates, composting, other sources of different organic residue in order to promote this biodiversity. There is a very strong meeting 7, 8 years ago. It was in Alabama. They took the most, not the most, 180 people. There was representing more than 60, 70 different experiments in the world. Rothamsted in England, East England, I've been twice there, 189 years now. They put together these people. It was Wayne Reeves that told me what's happened.
Very interesting. in terms of this.
To finalize,
Go ahead.
what did they achieve? That if you need to recover some soil, independent of the climate, the sandy, if the sea level or 3,000 meters, independent of the crop, the most important biodiversity, or says multiple plants, multiple species. When you put multiple species, more microorganisms, more times close, you recover this soil organic carbon, you recover the potential soil productive. We work with sugarcane, we have many different things in Africa, sandy soil. It's possible.
Yeah, that's
Biodiversity
definitely true. But you also pointed something that it's very important is that we need to know what is missing and what is and
Yes,
what are
the good
the
diagnosis.
farmers'
Yes,
targets in order to have more targeted
yes.
interventions, because we need
Perfect.
to actually ensure that the crop is productive towards the direction that we want. Thank you so much for your explanations. Also, we have Rocio here. Hello, Rocio. Rocio, we have a question here trying to understand a little bit more about your work. And the question is to when you mention your analysis to the microbial community of your rhizosphere, have you done matter by coding? I believe you have, because you also mentioned the 16S and the ITS, or have you done metagenomic? And in case that you have done metagenomic, did you identify microbial genetic traits associated to the different species or climate conditions? Can you clarify mainly what sort of analysis, metabacode
Yeah.
or metagenomic? Hello, Rocio.
Okay. Yeah. Well, we did both. In the project, I mean, we did both. Because in each of the plot we have 45 sampling points. In some of them we were analyzing the cystiness, and in other we were analyzing the metagenomics. Of course, cystiness is cheaper, so we were doing in order to identify which are the main the microbiomes, and then we were doing the metagenomics. The thing is, like, right now we are constructing the metagenomic assembly models in order to establish the metabolic pathways and these genome scale models to understand the metabolites. So we are just under constructing the models in order to identify, as Margarida put there on the question, this some microbial genetic trait associated. So we are currently on this way. I mean, performing the analysis. So I don't have the answer, but
The
yes, we
results
analyze
yet.
both. Yeah.
Okay.
Yeah.
Okay.
We are
Interesting.
currently
And you probably are also trying to associate these different traits with different species and climate conditions. And on this, and in terms of the experiment that you're doing, have you controlled also for the different farming production methods? So is it everything more conventional, more organic? Because that might also have an important impact.
Yeah, we have different also conditions. So here in Spain is conventional agriculture. Like in the north of Spain is like a normal practice that it is used for the wheat, but in the south, as I said, it is also included in some cases irrigation, which is not quite common for triticum, but considering the conditions. And in the case of Italy, it's organic agriculture. So we are also comparing the conventional versus So also, I mean, the experimental field that I show is what we did here, but of course also has been replicated in Italy, considering also the organic fertilization. So I mean, we are also comparing both.
Okay, just one final question, also for you, Admir, and Ieva, I don't know if you have work on this. When you're trying to measure this quality of your crops, and even in terms of the outcomes for the microbiome, do you test or do you assess the nutrient profiling of the food that you are producing?
Yeah, this is not so strong in Brazil, but many people know in USA. In a short time, I will go to USA. I've been many times in USA. I learned there a lot. And they are exactly many people, many research are doing this, evaluating the plants, the quality of the soil, the regenerative, and also the food quality. This is very important and more. I work also with some coffee farmers. They look, I love farmer. I love coffee. I love coffee farms. I was born in the coffee farm. And Brazil, the lady that I showed you, the Brazil, the ladies group is the most important regenerative coffee. It's a wonderful, with high quality, with the very minimal inputs, and some organics also we work is tremendous. Really, with the soil equilibrium, is less products, is unbelievable. This lady that you're working 4 years without potassium, without put, and the soil is more and more because they use permitted and other things, and soil life and all of these microorganisms is regenerating every season without putting the inputs. Because
Yes,
this is a
fantastic. And
good,
I'm sure
yeah,
I'm
the
sure we're going
alternatives,
to have some relationships also with the nutrient profiling. At
yes,
Food for
yes,
Sustainability, we are doing some work on that, and we
wonderful,
can already
wonderful.
see some relationships between the
Yeah.
practices and the nutrient. I don't know, Rocio, just one final comment. Is there something that in the project you have in terms of nutrient profiling of the the wheat in your
Sorry?
case? Nutrient profiling of the wheat.
Yes, sure. I mean, yes, we already also analyzed we are also analyzing the nutrient profile in the wheat, but also in the flour. So I mean, we
Okay.
are also milling the seeds. So we are also analyzing the quality of the flour, and also during this year they are going to do bread, and also in order to see the quality. So yeah, we are also analyzing. And I think what is the main goal, I mean, and what we are expecting at the end of the project is also how to see also this relation with the gut microbiome, and so that is we hope an interesting result for the project. But yes, sure, we are analyzing also the quality of the crop, but also the quality of the flour.
And those are very important results for us
Yeah.
to keep ensuring that we have enough arguments to regenerate, to bring more resilience to our soils, to promote the biosynthesis that Ieva was just mentioning to us. I think our time is up. Already we are over 15 minutes from our time. I want to thank you deeply for your time and for your insights, and sharing your expertise was a great pleasure. It was also a great pleasure to have you all, our participants, more quiet today, but it's always good to see familiar faces. I can see Annabella. And Annabella, nice to see you again. It's a pleasure. And just to remind you that this is just the start of our sessions, okay? So we'll be back next week with the smart tools for soil decisions. So we will be looking at how can we measure some of the aspects that we've been discussing here. So I hope you can join us. I hope you have enjoyed. And again, thank you so much for your presence here. So see you next week. Thank you. Bye-bye.
Thank you. Bye
Thank
bye. Thank
you.
you. God bless. Bye bye.
Bye-bye. Thank you.
Bye bye.
Bye.
Thank you.